So this video is going to go over the notes we took about the scientific method. First thing you need to know is that the scientific method is a method that we use for experimentation. It's used around the world, all different types of scientists, and not even just for science. The general steps that are used everywhere. And the point of it is to explore, make observations, and answer questions, and then through that, create a cause and effect relationship in nature. If I do this, then this is what's going to happen. So the purpose of the method is to figure out what causes different things to happen. So take a minute, and you can pause the video if you want, or you can just keep listening, but think about what are some of the steps that are included in the scientific method. Is there a logical order they have to go into? Do any of those steps have to be repeated? So just go ahead and pause the video for a second and think about that before we get into talking about the different steps. Okay, so hopefully you paused the video. So these are the general steps of the scientific method. First thing you have to do is you have to make an observation. You have to notice something. You have to see something or hear something or smell something in your world and observe, make that observation. Then you have to be curious, like you have to wonder about it. You have to wonder why it's happening. After that, we make our hypothesis, and we always do our hypothesis in if, then, because format, meaning if I do this, then this will happen because, and then give a scientific reason for why a certain event occurred. Then we get into the fun stuff. We get to conduct an experiment. After you have a hypothesis, you get to actually test it out and see whether or not you're correct. You collect your data and you analyze that so that you can draw conclusions. And then finally, what's most important is you report those results. No matter whether your hypothesis was proven correct or incorrect, you have to make sure that you actually report out what your results were. Now, even though this is a straight line, it should be said that in the real world, it's not a straight line. Oftentimes what will happen is a hypothesis might be proven incorrect, and I want to emphasize that that is when the real learning and the real science happens. When something goes unexpected, that's when we actually get to have those eureka moments and learn something brand new. But after those results are reported, you might go back and change your hypothesis and try and create a new experiment. Or you might retry your experiment. Maybe you think that maybe the methods weren't quite correct. Or maybe you have to look back at your data and say, did I analyze my data correctly? So even though this looks like a straight line, it doesn't always end up in a straight line. All right, now we're getting into some new stuff. This is the new information that you may not be as familiar with from science, but I'm sure you've heard at least a couple of these things in math class. The first is the independent and dependent. If you think about math class when you're making a graph, you have an independent and a dependent axes. In science, the independent variable is the thing that we change. So if I was looking at the growth of plants, the independent variable might be the amount of time I leave them in the sun. If I want to see how much sunlight my tomato plants need, I might have one tomato plant that's in full sun, one that only gets sun for a couple of hours a day, or one that gets very, very little sun. So the amount of sunlight would be my independent variable. The dependent variable, that is going to be what I actually measure, the data I collect. So if I go back to my tomato plant example, the dependent variable would be the height of the plant, or maybe how many tomatoes it produces, or how healthy it looks. That information that I collect, that's going to be my dependent variable. Now, in science and in math, the independent is what ends up on your x-axis, and the dependent is what ends up on your y-axis. So those two we're going to be working with a lot. We'll do graphing in another unit, but dependent is always y, independent is always x. The last type of variable are what we call controlled variables. And I know that sounds really confusing, like how can you control something that's variable? That sounds like they go against each other. But these are things that no matter which tomato plant I'm talking about, things that have to stay the same. In which case, there'd be things like I would need to make sure that they're both the same species. or that they are all the same species of tomato plant. I would have to make sure that they all get the same amount of water, that they all have the same size pot, that they're all the same age. Things that would need to be consistent from one sample to the next are what we call our controlled variables. Now, this is different from a control group. I know we got controlled variables and controlled group. They both sound very similar, but they are actually quite different. The controlled variables are factors, are things we keep the same, like environment. The control group is the group, the organism, or the sample that we use to make our comparison. Because remember, we're trying to determine cause and effect. If we want to see how something changes, then we have to have something to compare it to. So the control group is the thing that we are comparing to. So in this case, maybe it would be, I would have the tomato plants that is, my controlled tomato plant would be a tomato plant that is in my garden instead of under artificial conditions of sunlight. It depends upon the experiment, what your control group is, but it's kind of like you're creating your baseline. You're creating your normal environmental factors. So let's do a quick practice. Take a second, and again, you're going to pause the video, but I want you to think about this example. You have two groups of fifth graders, and they're tested to compare their speed solving long division problems. One group has calculators, the other doesn't. I want you to think, what is your dependent variable? So what data are you collecting? What is the independent variable? So what are you changing? And what are some of the controlled variables? So go ahead, pause the video and think about that. Okay, so our dependent variable, that would be the amount of time it takes for them to complete their long division problems. So that is what we are recording. We're recording how long it takes them to do. The independent variable, the thing that we change, is whether or not they have calculators. And then things that would be controlled, things that we would want to make sure were the same and consistent between both groups, is the age and the skill level of the students, the problems that they're given, the location they're being tested, and then of course the time of day, because our abilities and our focus, they vary depending upon the different time of day. So we'd want to make sure everybody was being tested at the same time of day. All right, last thing we want to talk about is a well-developed experimental design. Unfortunately, in school, we don't get to do all of these things because of timing and because we do an experiment and then we move on to new content and we need to move on to new experiments. But what we have, one factor that you need to have for a really well-designed experiment is many similar subjects. If you're testing something just once on one thinking about, again, our tomato plants, if I only have one tomato plant, one, I can't make any comparisons, and two, that one plant just might be either sickly or really healthy. So I would want to have a bunch of different plants in order to make sure I can take averages and things like that so that I have more accurate data. The second thing is you only want one variable at a time, meaning you only want to test one change at a time. So I couldn't change the amount of sunlight and the amount of water because then I don't know if my results are because of the sunlight or because of the water. So I'd only want to change one thing at a time. Next is the control group. As we kind of talked about, we need to have something to make a comparison to. If we don't have that control group, if I just have one tomato plant and I put it in this direct sunlight, then how do I know if that helped it or hurt it? Because I have nothing to compare it to. Last thing is that we must make sure that the test is reproducible. Just because something happens once does not mean that it will happen every single time. It could have been a fluke. So we want to make sure that we get consistent results over and over and over again. And we're going to be using this throughout the entire year, but especially really soon where you guys will be designing your own experiments. So think about these things when you get into that design group.